Welcome back to today's class where we're going to discuss these two questions you might have been asking yourself. So far, we have now just analyzed 483 and reduced it to 460 DNA binding proteins. And we've been using that word quite a bit, and we're going to take some time to talk about a little bit what those are, and then how we did some analysis already of what this data was telling us. And moving forward, we're going to use meta-analyses for some of our things. We'll go over what that means and what those plots look like. My favorite way of explaining a meta plot was defined by Professor Oliver Rando at UMass Worcester, and he calls it an averageogram. And that's pretty much what it is. It's the average of a feature. So what does the binding look like on a promoter on average for a DNA binding protein? So you kind of merge all of the data into one plot, and we'll go over what that looks like. Okay, so let's get started with what we're really trying to do here in class is sort of understand how 460 DNA binding proteins are sort of choreographed across the genome to define the cell state. In this case, the HepG2 cell state, or it's a liver cell. And so by the way these DNA binding factors can change throughout the genome, will produce the same genomic template, but where they bind will indicate whether it'll be a liver cell, in our case, a kidney cell, brain cell, or a heart cell, for example. And we know we'll also look at histone modifications that sort of barcode a given cell state. So where the histones are placed and how they're modified also signals to the cell what to be. And DNA methylation, we will not be analyzing this data in this class, but also can indicate what cell type you are. And transcription we will look at. So what genes are being expressed relative to these DNA binding proteins. We'll look at that in a later class on how to do RNA sequencing and compare where proteins are bound versus how much output is coming out from that gene. And really what you can think of this is we're looking at one sort of airport's how the planes are landing in one cell state, the HepG2. But as cells change, those DNA binding proteins change. And they then know how to define a new cell state. And this is kind of like air traffic control, if you will, that the genomic template doesn't know what genes to turn on. But the planes can be guided by DNA sequences that are recognized by these proteins to sort of traffic all the activity in the cell. So really you can think of it as there's thousands of planes in the sky and they don't really know where to go. And they need a signal in order where to land or to reroute, et cetera. That's really what we're looking at is 460 DNA binding proteins and how they're moving around the genome, how they're rerouted, et cetera, in one cell state. Okay, and the power of how these DNA binding proteins and where they bind was underscored by the Nobel Prize by Shinya Yamanaka, where he was able to put in 4 transcription factors into fibroblasts or skin cells that you could get from your body or anybody's body, and reprogram them into stem cells that can turn into any type of cell. And this held really great promise for medicine that you could take your skin cells, produce stem cells from them, and then change those stem cells into other cell types. And that's really been tons of work over the past decade or more in understanding how this choreography of all these proteins that we're looking at, or these DNA binding proteins, how are they coordinated and how does this result in gene transcription or cell state identification? And the more DNA binding proteins you have, you can see from these tracks, kind of like what we've been looking at, and we can get more and more information. So this is only a few. We're gonna have 460 lines by the time we're done with class. And so this promise has been realized that you can take skin cells, turn them into stem cells, and then you can use different sets of DNA binding proteins. So you can kind of like play the genomic piano by adding new proteins as you go. You play different notes to a song, if you will, being the cell state, and all the way to terminally differentiated cells. And so again, there's great promise that by understanding this code of how these DNA proteins are moving around the genome, what we're trying to do now is really important in medicine to find which transcription factors regulate which genes, which will then in turn change a cell state. This can also now even be done in 3 dimensions, where you can essentially, by using these long protocols, you'll see 15 to 30 days, there's much longer ones than that, but you can grow them in spinning flasks so that they actually grow into organoids. And if you look at these under a microscope, they look like the cortex of the brain and organized into the right layers as well. So this is wild type brain, and this is the organoid. So we can now sort of study biology, or organs, if you will, in a dish, mostly just because we started to learn how these DNA binding proteins choreograph specific cell states. And so this is the list from last year of 195 DNA binding proteins. This year we have 460. I didn't want to repaste all of them in, but you can see it's a lot of data. And we can start analyzing that, and we'll talk about that in just a second. But let's take a step back really quick and say, what are these DNA binding proteins, or how do they know how to bind DNA? And nature's come up with several clever tricks. This is one of the original papers sort of saying, here's types of protein structures that bind DNA and what those structures look like. And there's a few of them, and since this, we've known the structures of many, many more. But it still does sort of distill down into a few canonical ways of proteins being able to interact with DNA. And here's just 9 categories shown here, and we'll show a couple more complex ones in a second. But here you sort of have 2 proteins making like almost a tweezers around the DNA. And DNA is negatively charged, so these proteins are usually positively charged, so they can make sort of electrostatic connections and or read sequences inside the DNA. So some just bind via sequences, and some bind via these sort of like tweezer-like aspects. Or here in this example, you have a protein that's stretched across 2 different major grooves of the DNA, similar here with the helix turn helix domain. The immunoglobulin fold of an important transcription factor, NF-kappa B, in the immune response. And when you take aspirin, that it has a very bizarre fold that in the middle interacts with the DNA, one stretched out. As you can see, these are just basically how the different modalities of binding DNA. And so you can look for your favorite protein in the data set, or a protein that sticks out in one of our analyses, and we can ask, is this trend common across these classes? So this is already starting to think about meta-analyses, where we can say, here's the type of structures this class of proteins has. Did they do anything different than the other classes here? And so this is just more examples of the clever ways proteins have found to interact with DNA. With this beta zipper being kind of cool, where 2 helices just sort of do kind of like a molecular tweezers to the DNA. And we can think of this in 2 dimensions as well. P53 actually requires 4 versions of itself to bind to DNA. So each of these globs here is 1 P53 protein. And this bar graph here is just showing the domain structure of 1 P53 protein. Where you have a DNA binding domain. The transactivation domain is going to activate polymerase II to make expression when it comes in. So it'll sort of come in, bind to DNA, and push polymerase II to start activating. The DNA is here in the blue. You can see this tetramer is just kind of collapsed around the DNA. So it's very specific binding. And it binds a very complex sequence, which is actually a palindrome. So half of it is up here, and the other half of the sequence is down here, forming a tetramer. And none of these details you don't really have to understand, just that different proteins do have. So if we did ChIP for P53, we would retrieve this piece of DNA, but we would not know that there was 4 molecules sitting on it by the way we're analyzing the data. Some other ways DNA binding proteins find their targets is having a cofactor. So this guy, HMG protein, won't want to bind until it has its partner bound first. And then it can come in, or they can come in and find themselves through a dimerization domain. The same protein might do the same thing with a domain built into itself with a linker region here, and then clamp onto DNA with a helper site, and other sort of duality kind of ways of binding. So it can get complicated, and when we do ChIP, again, we're only going to see signal for one of these. and not quite know that that's there, but we can say, hey, what protein is that? Oh, it's an HMG. Was there another HMG cofactor there that might be dimerizing with it, etc. So all of this is really just to say that these are important classes of ways proteins can find DNA. And in our 460, since we have such a large number of them, we can actually subdivide them into these kind of categories and see if they have different properties where they bind in the human genome. It's also interesting to note that many of our DNA binding proteins don't have a DNA binding domain. And in ChIP, when we cross-link, the protein doesn't have to be directly bound to DNA. It could be, because of the cross-linking, we could be IPing the DNA that is bound by another protein that's bound to the DNA of the factor that we're looking at. Or there's domains that we just don't quite understand yet. And that these proteins are bound to DNA because they give signal in our ChIP assays. There's lots of resources. So when we're going through our analyses and you find a protein of interest, you can type like DDX20, for instance. You can type it into Uniprot. It'll give you a bunch of information that it's a helicase with an ATP binding domain. And you can start to see a lot of these helix turn, beta strand, DNA binding domains. But actually, DDX20 is also an RNA binding protein. So you can find out all that information in Uniprot. Similarly, the protein database has all of this information. Again, these two-dimensional sort of domain plots here of what domains are in this protein that you might have found in the data set. And you can view them in really pretty ways and print pictures of them here. So when we find something interesting or a DNA binding protein that stands out, one of the first lines of defense is to go find out more about that protein and see how it likes to interact with DNA. And does it activate transcription? Does it repress transcription? What else is known about it? And we'll do some of that as we go find some interesting genes. So that's our gene list from last year. Again, we have 460, so it's many more than that. But let's switch gears real quick to start discussing metaplots. We have 3 papers to read for the next class, but you'll see a lot of metaplots in there. We thought it'd be good to just take some time and sort of say, what is a metaplot and metagenomics? And we use transposons as a way of introducing them and how you can start building metaplots just like we will in class. So transposons are really cool because they comprise about half of the human genome. So this half is not transposons, which are essentially viruses that invaded the genome. They're different in all of us, but the percentages are about the same. And so these things have hopped into our DNA and may still be hopping, and we'll look at some of those. And what's interesting is species have species-specific transposable invasions, if you will. And we'll look at ones that are specific to the human genome, such as the SVA transposon and the ERV-H or LTR7 transposon. And we'll see why that could actually be of interest to us. Not to mention, one of the coolest scientists ever discovered them, and nobody really believed her, but she found these in corn, where corn kernels had different amounts of color in them. And that was because of hopping genes, jumping genes, as she called them, moving around the genome and expressing different colors in different kernels. And this is sort of a way for the genome to shuffle its deck in evolution and sort of move around. Or some people just think it's wasted space in the genome. But we'll take a look at it because we did find some interesting stuff last year and before. So if you look here, this is kind of busy. This is one of the papers to read for class. But really just want to point out one thing. This is not a metaplot. We'll get to that in a second. But if you look at how a transposon is enriched over the genomic average, and we'll talk about that. Because there are so many transposons, we have to have a null distribution of how often do they show up randomly in the genome versus the observation we have in class of how many times a given DNA binding protein overlaps a transposon. And we'll talk much more about that as we go. But basically, you want to take that into consideration. And what this y-axis is saying, if you randomly shuffle these around the genome, this is their enrichment for this feature. In this case, it's a long non-coding RNA that we talked about. in the previous video. And so we're saying in the bases covered of a long non-coding RNA, who's enriched more than average of the genome? And what you would see is that something that's covering a lot of long non-coding RNAs out here, and the more enriched it is over its genomic average would be up here. So these would be things that are just very specific to this feature that we're looking at. And what we and several other groups noticed quite quickly is this ERV-H int. ERV stands for endogenous retrovirus, H stands for human, and this H stands for how it's reverse transcribed, which is not important, but a histidine tRNA will use it. It's also known as LTR7, so that's here. It's a subclass of it. And so the fact that both of them were there was quite striking. So if you look closer at where they were, they only overlapped with long non-coding RNAs and not mRNAs. And that was the first surprise. And we saw in the previous video that we're going to do a lot of that comparison. What's the difference between an mRNA and a long non-coding RNA promoter with respect to our data set? But what was interesting is this long non-coding ROR, which is important for that reprogramming process we talked about, Yamanaka's discovery, where when you put in those pluripotency factors, link-ROR, or this long non-coding RNA, is actually one of the first genes turned on, and that was actually discovered by Yamanaka himself. And it has this ERV-H transposon in it, which is only in humans. And it's right, these LTRs are essentially promoters. They're viral promoters. Called, the LTR stands for long terminal repeat. And what we can see is it's right at the start of this gene. And that was quite striking because we knew this gene was important from our studies in Yamanaka's. And it's surprising that it would have been created. It's only in humans. It doesn't exist in mouse. And it's surprising that it could have possibly been created by a human-specific transposable event. But what was interesting is that there was 127 examples just like this one that we knew was important. They all start right there, and they're all in the same direction. There was a 50-50 chance that this LTR was pointing the other direction, and a gene would have been made in a different way. So they all were essentially making promoters. And this is where we wanted to get into meta-analysis. So we have 128 observations of the LTR possibly promoting a non-coding RNA, and we have thousands that are not. But we want to look at what's unique about these 127. So we want to average them rather than look at all of them individually. So let's see what that looks like. Well, I guess one side note is of interest is of those 127 LTR7 genes, they're only expressed in pluripotent cells. And that's interesting because link-ROR, as we said, was important in that reprogramming process into a stem cell. And is it expressed only in stem cells, only in human stem cells, that all of them were like that. And that's really surprising because stem cells don't like to express transposons. In fact, when Yamanaka first did the reprogramming experiment, he added a virus that contained the 4 transcription factors to turn a fibroblast into a stem cell. And the way he defined reprogramming as complete, or a complete pluripotency cell, actually put DNA methylation to silence the LTR7, or the LTR of transposon, of the virus that was used to reprogram the cells. So stem cells love to methylate anything that looks like a transposon. And that's really important for them to be silent in a stem cell. Or that was what commonly was known. And this was an example of a strong exception that if you are an ERV-H or LTR7, you are specifically expressed in a stem cell, unlike almost every other family of transposons. So that's interesting. And now we can start to sort of split these two into groups and get meta, if you will, or average out them. And one of the, it's not really a meta plot, but you could think of it kind of this way, is a cumulative distribution frequency. And we had a little exercise in the last video of deriving an empirical cumulative distribution function. But just to recap, what this is doing is saying, where does your data saturate to 1? At what point have we represented 100% of the data? And so here you're seeing 2 plots where the expression of the gene, so how much of the gene is being expressed, and between 2 different groups. And we'll see. So that's sort of a meta plot. We've grouped them into two different aspects. Or as Ollie would say, the averageogram. We've averaged out, we don't see it in an individual data point, but we average out these categories. So red is if you have an ERV-H, what is your expression in a stem cell? And if you don't have one, what is your expression in a stem cell? And it's very clear that if you have an ERV or an LTR7, you're going to be expressed very highly in human ES cells or human embryonic stem cells, whereas the distribution of expression of non-ERV-H saturates much faster or has much lower expression levels. So that's quite surprising, and it fits with this data here, where red is sort of indicating more expressed and white is less expressed. Okay, so could this be that this LTR7 actually contains pluripotent binding sites for OCT4, SOX2, and NANOG, or these magic transcription factors that can turn a cell into a stem cell? And so here's a meta plot where we're going to take the start site of those 127 long non-coding RNAs, and we're going to look at ChIP-seq data, just like we are in class, and we'll make these meta plots in about 2 more classes, where we say, okay, in the TSS of an ERV containing or LTR7 containing region, this is where we're going to call the start site. Where is OCT4 bound relative there for all of those 127? And so what we can see is that it actually binds right at the transcription start site. That's a strong indication that perhaps OCT4 is part of the reason why these ERV-H or LTR7s are expressed in stem cells. And so we can look at that now a little more relative to other transposons. So let's look at now a triple meta plot, if you will, where we just saw this curve where OCT4 likes to bind right at the top of this, right at the promoter or at the tip of the LTR7. But if you look at other families like signs and lines, they don't do that. So this is the average of thousands of these elements and where they reside next to ERV-H start sites and whether there's OCT4 enrichment. And if OCT4 was enriched at all, it'd be out here and nowhere near where the start site of a sign or a line was. Whereas if it's an ERV-8 start, the ERV1 start, then it's a LTR7 start, then it's enriched. And so what we can see here is NANOG is also there. So this is the meta plot, chip fold enrichment at the transcriptional start site of an LTR7 for NANOG. And again, we can see it's starting almost just a little bit to the left of where we saw it for OCT4. And that would be that's double information that this transposon has these motifs in them to bring these important proteins there, but they're right next to each other. And that's more convincing that there's some biological reasoning for that. There's been tons of reviews about this since, and you've read the paper, but really it's been found by many, many groups that these LTR7s are not only expressed in human stem cells, but also in the very early stages of fertility and implantation, and they're important for the human embryo implantation. So you have to wonder, how do mice and humans do the same things as different species? And perhaps some of these species-specific transposons are tweaking some of the needs of individual species. And there's more papers are coming out more and more about how these LTR7 regions are shaping enhancers and other things, not just the promoter regions of long non-coding RNAs. And so now we want to go see, with this much larger data sets, what proteins prefer to bind to these guys. And we did find some interesting things last year, and I'm guessing we'll find even more with having this many more proteins. So with that, we can see what the sort of more typical metaplots are going to look like. So we're going to use this, unfortunately, nomenclature interchangeably. TF stands for transcription factor. It's also a DNA, these are DNA binding proteins, but they're one subclass of them. So really DNA binding proteins covers everything in class. And so what we can do is make a meta plot shown here, like let's say this middle part here is a promoter. And what you're saying is these are OCT4 ChIP sites, and each line is a promoter. And the center is where the transcription start site is. And so as we saw in the previous plots, let's say this guy starts right in the middle, and you can see the dark blue signal means there's binding, and the light blue means there's not binding, indicating that this thing binds right in this position. And across all promoters, it does that. And that's why it's important to look at a meta plot, as you can see how consistent this pattern is. And here's another meta plot asking where the histones are relative to that. And you can see it has sort of two patterns here in it. And but now again, each row is a promoter, and where the binding activities in deeper shades of red. And we're going to make some of these profile plots as well. Then another meta-analysis is you can say, well, these regions that were bound with in the red and blue, but not down here, you can see that the binding motif for OCT4 is enriched, validating that. Really, this is the same thing for SOX2 shown here. Okay, so these are sort of heat map meta plots where you center something and you can't really see an individual gene, but you can start to see the patterns of different categories within the gene. For example, these have no binding activity down here and down here, but these do have binding activity of histones that package DNA. These are just more meta plots. This is showing how you can do this for RNA-seq, which we will also do. Here, the red strand is the plus strand and the blue strand is the minus strand. These are what are known as enhancer RNAs. And you can find where a transcription factor, DNA binding protein, is right in the middle here, right where these peaks shift from the little RNAs coming from the left and from the right. And you can see this as a meta plot here that some promoters have it very strong and then some are quite weak. And then you can sort of look at this metadata and ask what's enriched in there. And this is all very busy. It's just another example that you can do this for RNA-seq, ChIP-seq, or anything, where you sort of set a position and then say, what's the trend of every observation as a row here. You're kind of averaging the data in a way. Here's another way to positionally reference, and we've already started to do this with our ChIP data, where we've said, well, let's look at just the promoter and the gene body. And we can also get the UTR of a gene if we wanted as well. And just to show that this works for not only ChIP, which is asking which pieces of DNA a protein is bound to, but CLIP is used for finding the RNAs that an RNA binding protein is bound to, which is essentially equivalent to what we're doing. And we will make plots like this as well. And so you can see here, there's certain proteins that prefer to bind to the 5' UTR. There's another set of proteins that prefer to bind to the middle, and there's another set of proteins that prefer to bind to the outside. And we've already started to see some of this in our data, and we're going to see more of it, of DNA binding proteins that prefer to bind to the promoter versus the gene body, which we already kind of started to see. And this is just kind of a meta plot here, where you can show the positions of things and then the results of your data. So let's say you ran a statistic and said, these RNA binding proteins bind to the 3' UTR. Well, you'd expect the average signal for that group of genes to have its peak near the 3' UTR. And that's exactly what you see. This has coding sequence or gene body enrichment. That's what you see as a meta plot. And you can see how they're really helpful, because you can start to see the trends without knowing all the data for every individual gene. And so that's kind of what we mean by meta plots, and we'll make very similar plots to this, but with ChIP data, and talk more about the shading scales, which indicate binding activity in low and high and different color schemes we can use. So with that, let's go start making, moving forward with our analysis. We made all our consensus peaks, we analyzed the global properties, and we'll start moving forward in producing meta plots next, and seeing what are the average trends of certain classes of proteins, or what does the binding look like at promoters? Are there unique binders? Are there ones that don't bind to promoters, etc.? we're going to start doing meta-analyses of lots of different genomic features. So until then, cold light, and thank you.